So I'm going to tell you about the Gene Wiki. This is another Wikipedia-based initiative. And I'm going to go pretty quickly through the background and some of our published data so we can get to some of our recently published and unpublished data. To describe sort of the overall motivation for the Gene Wiki, it really evokes this concept of the long tail. So for the people who aren't familiar with the long tail, let me explain. It's best explained first by looking at its opposite, which is the short head. The short head is a situation where you have a few number of individuals, each creating a large amount of content. And I hold the example here to be newspapers. So there are about 1,500 daily newspapers in the United States. The long tail is the complementary resource. That is where you have a large population of individuals, each contributing a relatively little amount of content. Okay, so and here I hold up blogs. So while there are few blogs, if any blogs, that rival the amount of content being produced in a newspaper, in aggregate, the blogosphere, which is millions of blogs online, clearly rivals, if not outpaces, sort of the amount of content generated by newspapers. This hits many other areas of society, comparing YouTube versus TV and Hollywood studios, Consumer Reports, the short head version, the long tail version of that is Amazon reviews, and so on and so forth. The point of the Gene Wiki, behind the Gene Wiki, is to extend this idea to gene annotation, right? The short head of gene annotation is manual curation, right? We have several centralized groups of professional curators whose job it is, and they do a fantastic job, and they each generate a lot of content. And the question is, can we engage this long tail of scientists, i.e. the people in this room, the people in the labs, to directly participate in this process of gene annotation? And that's fundamentally the hypothesis behind the Gene Wiki project. Can we harness the long tail of scientists? So the most successful long tail application is undoubtedly Wikipedia, as Alex introduced earlier. There are only two things that I think are relevant for you to know. And first, that Wikipedia is relatively accurate. This is a study done in Nature several years ago that showed that it was comparable in accuracy to the online Encyclopedia Britannica. The other thing you get from Wikipedia's open infrastructure is incredible breadth and depth, right? So if you compare Wikipedia versus Britannica Online, both in terms of the number of articles and the number of words, these are sort of some older stats. I mean, far and wide, Wikipedia outpaces things like short head types of resources like Britannica Online. So it has incredible breadth and depth, and it also is reasonably accurate. So what we thought to do is to engage the user community in this process here, moving from a document-centric view of the world of biomedical information down to a concept-centric view of the world. And what does that mean? Well, if you're interested in a concept, for example, fibronectin, a gene that maybe popped up in your microarray study, and you go to PubMed, right, you can wade through 30,000 different articles that are relevant to fibronectin. But it would be much more convenient to actually crowdsource the process of filtering this data and summarizing it into a single Wikipedia article that summarized essentially the relevant facts that were known about fibronectin. So how did we approach this? Let me start with this. I think there's one principle that really underlies all successful crowdsourcing application, and it's this positive feedback loop that I'll describe here. The first thing we had to do is establish a Gene Wiki page that had some baseline level of utility for the community. And that baseline level of utility would attract some number of users over some amount of time. Some percentage of those users, even a small percentage of those users, will actually stay to make a contribution. And that contribution could be as trivial as fixing a typo. It could be as substantial as summarizing a recent paper in the literature. But in the process of making that contribution, right, it makes the page that much more useful, which then in turn attracts that many more users and attracting more contributors and so on and so forth. So it's this positive feedback loop that I think underlies the Gene Wiki and it underlies many other crowdsourcing and community-based applications. So our approach for developing the Gene Wiki was very similar to what Alex described, is that we also created stubs. Initially we created close to 10,000 stubs within Wikipedia. And these stubs were based on data that were mined from structured databases that are already online. So gene summary from NCBI, protein interactions from BIND, I believe, references from NCBI, gene ontology terms, and so on and so forth. And by creating this systematic and baseline level of content for close to 10,000 pages, this provides a useful resource to the community. So this is establishing the first leg of that positive feedback loop. This is what establishes the utility. So how are we doing in terms, so I'll say that this is similar to what Alex did and his team did. How are we doing in terms of users? Well, users, again, as Alex mentioned, just by being in the fact that you're in Wikipedia, you get a lot of visitors. So in aggregate, the about 10,000 pages we have in the Gene Wiki are viewed over 4 million times per month. And there's a wide distribution shown here. About, I think, 80% of Gene Wiki pages show up on the first page of Google when you visit them. So I think it establishes that we have a critical mass of users and are poised for future growth of users. In terms of contributors, we get, let's see, about 1,000 edits per month from the community, sprinkled over all 10,000 of those articles. So on a per article basis, not particularly high, but aggregated over the entire Gene Wiki, we think it's indicative of a strong critical mass. And comparing productive edits versus vandalism, I think we're doing pretty well there. So I think this establishes the critical mass. And just to show what could be the result of the Gene Wiki article, this is the article I like to show is what we could have for every gene in the human genome. This is the Wikipedia page for a gene called relin. It spans 6 screenshots here. It starts with a pretty nice table of contents spanning all sorts of different topics. It has figures and diagrams here showing expression of relin in the mouse brain. Here's a section on the pathological role of relin. 2 critical things to highlight here. First, there are inline citations to the primary literature. So here, if you click on this link for reference 104, it goes directly to this full citation with 1 click away to the PubMed reference. And the second thing is that if you're not familiar with any concept, for example, Norman-Roberts syndrome, you're 1 click away from this broader ecosystem of relevant biomedical information. So these, I think, are all things that make the Gene Wiki pretty useful. To underscore the point, this is a community-based initiative. relin was, this page that I highlighted was created by almost 100 unique editors, over hundreds of edits over many years. Here are some other cherry-picked, biologically relevant articles that have very similar patterns. So I think it really is a community resource. So if we stopped here, and this was initially our intention, just to stop here, what would we have? We would have roughly 10,000 articles that would be great for humans to go read, right? And that would be fantastic. But what we realized is that we, as the informatics community, right, are interested in making the Gene Wiki more and more computable, so we can integrate that knowledge into our analyses. So what have we done? We've taken first structured annotations from structured databases online. We've fed that and reformatted that into Wikipedia pages. We've invited free text contributions from the community. And now our goal is to complete this loop and convert these community contributions back into structured annotations so we can do all the bioinformatics analyses that we know and love, from pathway analyses to enrichment analyses to cross-genome comparisons and so on. So how are we going to do this? Let me illustrate one very basic mining methodology. I won't go into too much detail. It's published here. But I'll highlight this example here. So this is the Wikipedia article for the 5-HT6 receptor. And it starts with the sentence that's the 5-HT6 receptor is a subtype of the 5-HT receptor that binds endogenous serotonin. So there's the serotonin receptor. And the 5-HT receptor, right, that text is wiki linked to the article on 5-HT receptor, what it means to be a serotonin receptor. So we know that this article corresponds to this particular NCBI Entree gene ID because we had a role in creating that article. And we can also use the NCBO annotator, just essentially doing basic concept recognition, to know that the 5-HT receptor actually refers to serotonin receptor activity corresponding to this gene ontology term, right? So based off of these mappings, you might assume that there is a candidate assertion here. Okay, so 5-HT6 receptor matching to serotonin receptor activity might not be terribly earth-shattering, but in fact, that is a gap in the annotation, right? That annotation actually does not exist in the database. Again, I checked last night. So this gap in the annotation, right, could easily be filled from annotations that are mined from the literature. Again, to highlight the initial text, we can go to the inline reference that was cited here. This is the original cloning paper from 1996 describing the serotonin receptor. So it would be pretty simple for a professional curator to translate these findings into a formal annotation that could again go back into the structured annotation databases that then again we could compute on. I'll just do one more example. This is the gene for APLP2, a clear link to endocytosis. Endocytosis has this gene ontology term. APLP2 has this NCBI entree gene ID. This is a candidate assertion. A little bit of literature searching later, you can easily come along this paper that talks about the role of APLP2 in endocytosis. So it's this pattern that we've gone through. Through this pattern, we mine out about 11,000 novel annotations from the Gene Wiki. We compare that to existing databases, the existing Go annotations, and about 43% of them match known annotations. And that means we have about 6,000 novel annotations. In work that I'm not going to describe here, we peg the specificity of these somewhere between 48% and 64% specificity. So that is not particularly impressive, but let me describe what the follow-on that we've done with that. The question is this corpus of 6,000 novel annotations with modest specificity useful in the context of these 100,000 annotations that are already available from the Go consortium? And so we devised a little analysis to test that. The analysis focuses on this idea. So let's start with a Go term, and the Go term, we can go to a particular gene list. From that gene list, we can search in NCBI and get to a list of PubMed abstracts. Okay, so now we're in free text land. If we forget for a second how we got to this gene list, we should be able to go to these PubMed abstracts. We should be able to do concept recognition, do enrichment analysis on relevant terms found in these PubMed abstracts, and we should be able to get back to the Go term that we originally started with, right? This would be the nice gold standard. I'll illustrate how this actually works. So Exon Guidance, there are 264 genes available for Exon Guidance. That maps to 811 articles for these 264 genes. And of these 264 genes, 13 of them are linked through PubMed to the concept of Exon Guidance. Okay? So 13 out of the 264 relative to the background rate for Exon Guidance actually turns out to be incredibly statistically significant. So this works out absolutely great. But let's take a look at another analysis where it doesn't work out so well just based off of PubMed abstracts alone. We can look at a term for muscle contraction. There are 87 genes here, mapping to 251 PubMed articles. Of these 87 genes, none of them actually are linked through PubMed to this term for muscle contraction. But if we add to this the Gene Wiki mined articles for these 87 genes, what we actually find is that the Gene Wiki articles for 6 out of these 87 genes actually explicitly mention muscle contraction. And this, of course, leads to a very significant p-value that is shown here by that 2x2 contingency table. So I've shown you one example here looking at muscle contraction. And if we were to plot the p-value for PubMed only versus PubMed plus Gene Wiki, the more significant things are down in this corner, less significant over here. Things that are benefited by adding Gene Wiki content fall on this side of the plot. Muscle contraction is there. And now if we run this over a large collection of gene ontology terms, you can see that by and large we are overall improving the state of enrichment analyses through these novel annotations mined from the Gene Wiki. So we think this is a pretty nice demonstration that the Gene Wiki can be a useful resource for structured gene annotations. So what I've described here is, so far, is that we're getting to good text that humans can read. We can also mine that text for annotations that are useful for enrichment style analyses. And now the next question is, how do we disseminate this information to the community? So there are a few different strategies, and let me describe one that we're pursuing right now. We're actually created a system that we call Gene Wiki Plus. Gene Wiki Plus is a semantic media wiki instance, for those who know what that means. And essentially, we're syncing content between the Gene Wiki into our Gene Wiki Plus system, and where we're doing our basic concept recognition mapping I described here. We're also bringing in Snipedia. So Snipedia is an analogous program for annotation of SNPs. And what we can do now then is actually do dynamic queries across both of these crowdsourced initiatives. So now we have the page in Gene Wiki Plus for AKT1. And at the bottom of the page for AKT1, we can see, for example, all of the SNPs that are known for AKT1, as well as the related phenotypes from Snipedia, or the mined diseases out from the Gene Wiki pages. So these are now structured annotations that can be automatically displayed against these pages in Gene Wiki Plus. Because Gene Wiki Plus is a semantic media wiki instance, we can start to do interesting visualizations and analyses. Here's one visualization that we're developing with somebody in the context of the Google Summer of Code program that Alex mentioned. We can also do broader visualizations like this, where we're looking at linking the top 100 genes to various diseases and drugs, these biomedical concepts, as well as the editors who made the contributions. And we can also do integrative queries now based off of the Gene Wiki Plus content. So for example, the Semantic Media Wiki software over here allows this type of dynamic query and integrative query. So we can essentially look for all human proteins where they are associated with breast cancer or has a SNP that is associated with breast cancer. And from that, you can generate a list of genes in programmatically accessible way to do that query. And that's also to say that if we add now, for example, AKT1 is related to leukemia, for example, on Wikipedia, right, in a very short amount of time, that would be reflected in this type of dynamic query. We can also imagine add many other resources here. So if we, for example, have talked about with Alex Pico, we could also sync in Wiki Pathways content. So you could add the context here of and is involved in the apoptosis pathway. We could also add other databases that are non-media wiki based that we want to access through this type of syntax directly into Gene Wiki Plus. So Gene Wiki Plus gives us a mechanism to do some queries here directly within there, but we also want to export into RDF. It also, the Semantic Media Wiki software allows this native export into RDF, and RDF, of course, gives you integration with the broader context of linked data, so you could query across many other resources as well. So this gives you a sense for where we're going in terms of the dissemination as well as the analysis opportunities that the Gene Wiki mining affords us. I hope I've convinced you that the long tail of scientists is a valuable source of gene function. Crowdsourcing and community intelligence is one of the themes of our lab. I'll just highlight in one slide that we also have our crowdsourcing a gene annotation portal called BioGPS, essentially looking at distributed numbers of gene-centric resources that you can mix and match in your own customized layouts. Nobody should leave this room for the next talk, but if you do, my colleague is giving a talk on this in one of the other, I think in the technology track. With that, I'll wrap up and thank the people who are involved. These are my group members. These are, in particular, the people on the left-hand side have been instrumental in the work I've described here on the Gene Wiki. This is a long list of collaborators, including the Molecular and Cellular Biology Project, I want to especially thank Biomed Central for travel supports for me and my lab to come to this meeting. These are the funding sources and how to contact me, and I'm happy to take any questions. Thanks.
Hi, so your semantically enriched annotations, they are not in Wikipedia, but in your own kind of database or something like that, right? Can you comment on why they are not in Wikipedia, and are there any chances in the long run in Wikipedia will support those there?
The question is what Gene Wiki Plus is housed on?
Yeah, like basically, I think your semantically enriched annotations are very valuable.
Right.
So it would be great to see those in Wikipedia. So I was curious, why can't you do them in Wikipedia, and why is this outside?
You're asking DBpedia, is that right?
Wikipedia.
Wikipedia.
Yeah. If I understood you correctly, Gene Wiki Plus is outside of Wikipedia.
Yes. Yeah.
Yeah. So.
Okay. So it is a sync of the Wikipedia page, right? So we actually don't allow editing of the text on Gene Wiki Plus. What happens is that if you edit things onto Wikipedia, those in roughly real time get synced over to Gene Wiki Plus together with the concept recognition that we do. Obviously, if we forked completely and allow editing over here, we get into weird synchronization problems, and that's something that we did not want to have. So essentially, the Gene Wiki paradigm of combining Wikipedia content with Snipedia content, those are one-way syncs into our system.
Okay.
Does that answer your question?
Yeah, thanks.
Slightly left field question. The information that you're pulling in from Snipedia, I'm just thinking about that in the context of consumer personal genomics. Do you think there's a lot of, I don't know whether risk is quite the right word, but perhaps opportunity for less specialist audiences to derive useful information from what you're doing?
It's a good question. So I should say that the Snipedia developers, right, that is their goal. That was their goal in creating Snipedia, to try to reach the people who are accessing consumer genomics services and so forth. So, so I think they have software underlying it that allows you to interpret your consumer genomics data. I think integrating with GeneWiki certainly does allow for a richer ecosystem of information, because then we can relate to specific genes and other concepts. But absolutely, I mean, I think the goal of our initiative is to essentially try to get our data into a structured format so that other people could build innovative tools on top of the combined data set.
I was interested so you mentioned the number of annotators that you had for a couple of your examples. What's the distribution in terms of the is there typically one main annotator per gene? And can you tell sort of where that person might be within their career? Are they mostly students or postdocs, or sort of, and how long does it really take to annotate sort of the typical, I mean, are we talking hours, months?
It's a full range, and I think we don't have good hooks into getting more demographic information about the editors, right? Editors are essentially defined by their handle, right? And so for those who saw Alex's talk, you'll recognize BogHog, okay? So BogHog is one of these guys that edits a ton of different things. We have a poster-sized version of this I wish I brought, but if you notice what BogHog does is he actually edits many of these genes. In contrast, you see other editors who are essentially single-issue editors, right? So there are people who only edit this gene. I think, I forget which gene, this is some sort of receptor. There's one person, essentially, who's responsible for most of the content behind Relin, that gene I mentioned before. So we can get some basic editing patterns in terms of the number of articles that each editor is editing. In terms of where they are in their career path, I know we have many students. I know we have many professional scientists in biotech and in academia. it's hard to get firm numbers on those sorts of things.
Is it possible to tell how like to get reland, which is the good one, like was that hours of work? Or like how long does it take to get quality?
Yeah, so you can actually go back into the history of any Wikipedia article and you can see essentially how clustered the edits are. You get a date stamp on every single edit. So you can see that, Copper Kettle, who happens to be the person who edited this Reland article, has been working on it, tinkering with it for months, if not longer.
So you said you seeded WikiGene with about 10,000 stubs, which is heroic. And I'm curious if it's grown beyond that into other proteins, and how you keep track of which articles in Wikipedia are useful to you and which are just random pieces of pop culture.
Yeah. So essentially, we define anything in this informal collection of the Gene Wiki as anything with this info box over here. This essentially is what identifies it as a human gene. And this is something that we have tools that you can automatically create it using a simple web page that we've developed. I should say that we wanted to create all 20, 25,000 human genes, but Wikipedia has this concept of notability, and in one of the concessions to the Wikipedia community, who didn't believe that all 25,000 human genes were notable, we said we'd take the top 10,000 that had sort of the most human references. Yeah. Does that answer your question? Yeah. Great. Thanks. Oh.
We have time. So we have 5 minutes before the next talk. Any more questions?
Great. Thanks.